Good evening everyone. How's you? I hope you all are well. I am here to discuss a topic that is the mitochondria and chloroplast. The question has been asked in UPSC paper 2 that semi-autonomous organelle. That is the semi-autonomous organelles. Question was asked in 10 marks. So semi-autonomous organelle, what does it mean? Semi means half, autonomous, they regulate themselves, means they have their own ribosomes, means they form their own proteins, as well as they have their DNA, circular DNA. So, in a cell, in a eukaryotic cell, mitochondria and chloroplast are the semi-autonomous organelles. These are the semi-autonomous organelles. What are these? That is the mitochondria as well as the chloroplast. So we will study upon the mitochondria and chloroplast कि क्या है ये दोनों. Firstly हम similarities देखेंगे कि mitochondria and chloroplast में क्या similarity है. Both have their circular DNA. Yes, both have their circular DNA. Both have their own ribosomes, that is the 70S ribosomes, and both are the double membranous organelle. क्या है दोनों ही? Both are the double membranous organelles. Yeah. Yes. And there are double membranous organelle means two membrane is present, outer membrane and inner membrane. When we say outer, so outer they have the proteins that we call it as the porins. Porins for the exchange of the material from inside to the outside, and it is permeable and inner membrane is selectively permeable. Yes. Now, let's start with the mitochondria. And these both are only divided by the binary fission. Binary fission. So, I hope you got the idea that how mitochondria and chloroplast are similar to the each other. Now, first we have the mitochondria. Mitochondria. We used to say that mitochondria is the powerhouse of the cell. Yes. Why? Because it forms the ATP during respiration. So it is a double membranous organelle, double membranous bound organelle. And the amount of the quantity of the mitochondria differ in every cells. How? More the activity of the cell, more will be the mitochondria. Then activity differs. Means depends upon the cell. If cell is performing more metabolic activity, more will be the number of the mitochondria in a cell. It could be 1, 2, 3, many. Now, exception is in mature RBC. In mature RBC, mitochondria is absent. These are absent in the mitochondria. If we say what is the location of the mitochondria, location. So location of the mitochondria is in the cell, that is the cytoplasm. What is that? It is a cytoplasm. Mitochondria you cannot observe easily, so you have to use the dye, that is the Gyanaj dye, to observe the mitochondria. You have to use stain, that is the Gyanaj stain. Yes, as well as it shows the endosymbiosis with bacteria. It shows endosymbiosis with bacteria. Endosymbiosis means it shows resemblance with the characteristic of the bacteria. So we can say that mitochondria originated from the bacteria. We said that okay, it is a double membranous organelle, outer and inner. So let's discuss with these two. Double membranous, first is the outer, second is the inner. Now the outer membrane. So the outer membrane is smooth. It is permeable. On outer membrane, proteins in the form of porins are present. Porins are present for the exchange of the material from inside to the outside. And between the inner and outer membrane, we have the intermembranous space. What is that? Intermembranous space. And the inner membrane, here on the inner side of the membrane, we have the proteins are present. On the inner side of the inner membrane, we have proteins are present. Means proteins are made up of the some complexes. 5 complexes that helps to formation of the ATP, that forms the ATP through electron transport chain. Is that clear? Yes. Now this inner membrane is not smooth. Here foldings are present. What is that? Foldings present, and these foldings we call it as the cristae. What is that? Cristae are present. Now, what is the function of the cristae? They increase the surface area. What is the function? Increase surface area. And this inner membrane is not permeable to the all molecules. It is very impermeable to the solute. Inner membrane is impermeable. Now why it is impermeable? Due to the presence of the phospholipids. Presence of the phospholipids. That phospholipids we call it as the cardiolipids. What is that? Cardiolipids or cardiolipins. Now when we talk about the inner membrane of the mitochondria, so let me draw here. This is your outer membrane. This is the inner membrane. Yes. Outer membrane and inner membrane. Now these outer and inner membrane is made up of the proteins, lipids, plus carbohydrates. But the quantity of proteins and lipids vary in outer and inner. If we talk about the outer, so outer contains 40% of the lipids and 60% of the proteins. When we talk about the inner membrane, so 80% are protein and 20% are lipids. And this space is the intermembranous space. Now this is known as we call it as a cytoplasm, but in mitochondria we used to say it is a matrix. In matrix, what is present? Ribosomes are present. What is present? Ribosomes are present. And the ribosomes are of 70S, that is present in the bacteria. 70S. Now, the mitochondrial DNA contains circular double-stranded DNA. What is that? Circular, not a linear. If we talk about the eukaryotic cells, it contains double-stranded linear DNA. But in the mitochondria, that we have the circular double-stranded DNA. And not one, it could be many. It could be many. Circular double-stranded DNA. Now, the inner membrane have two phases. One is the N phase and one is your P phase. This one is the N phase. Means cytosol phase. Why cytosol? Because it is projected towards the cytosol. And this is your P phase. P phase, that is towards the not cytosol. It is a matrix. Yes, it is a matrix. With the matrix, it is a cytosol. Yes. Now, on the inner membrane, we have the enzymes are present. That enzyme that we call it as the ATP synthase. What is called? ATP synthase. ATP synthase, that is in the complex 5. That is present in the electron transport system. And all the ETS runs in this inner membranous space. And when we used to study about the Krebs cycle, so one enzyme that is present in the inner membrane, that is the succinate dehydrogenase, that is also known as the complex number 2. That also helps in the ETS, electron transport system. Means electrons will move and proton will shift, and this leads to the formation of the ATP. Yes. So this is all about the mitochondria. Now there is a portion of the genetic portion that how the non-nuclear DNA is inherited. How non-nuclear DNA means we are talking about the mitochondrial DNA is inherited. Mitochondrial DNA is inherited from parent to progeny. So I will give you just gist about this. I will give you gist about this. Let's start with the what is else could be present in the mitochondrial matrix. So it contains multiple copies of the double-stranded DNA. Double-stranded DNA. उसको करने से पहले हम पहले ये कर लेते हैं कि मैट्रिक्स में और क्या क्या चीजें प्रेजेंट हैं, as well as the ribosomes that I told you, that is the 70S ribosome present है. tRNA present है, various type of the tRNA is present है. tRNA, that is the transfer RNA, and various proteins present है. And as well as we calling it as a semi-autonomous because it has own genetic material, own ribosomes. Ribosomes क्या कर रहे हैं? प्रोटीन बनाएंगे. प्रोटीन that is the and mitochondrial proteins are encoded by the nuclear genes. Mitochondrial proteins are encoded by nuclear genes. So genes क्या है? Genes are the hereditary factors हैं. Means nucleus में क्या present होंगे? Genes present होंगे और वो क्या करेंगे? mRNA बनाएंगे. mRNA then comes into the cytosol. cytosol में क्या होगी? ट्रांसलेशन होगी. ट्रांसलेशन से क्या बनाएंगे? प्रोटीन बनाएंगे. And these proteins will be imported into the mitochondria. Mitochondria. And this mitochondria organelle is dynamic, means it frequently divide and change its shape. It is a dynamic. Frequently divide and change shape. And how it will divide? It will divide by the binary fission, just like a bacteria. So according to the endosymbiotic theory that is proposed by the Lennon, they said that they evolved in the eukaryotic from the endosymbiotic association of purple photosynthetic bacteria about many years ago. So how this mitochondria evolved? For example, I am taking the ancestral prokaryote. Now what happened in this ancestral prokaryote? There was in membrane enfoldings, inner membrane enfoldings. क्या हुआ? inner membrane की folding हुई. inner membrane की folding हुई, that is the enfolding of the plasma membrane. Enfolding of plasma membrane. After the enfolding of the plasma membrane, what happened? There is a association of the aerobic bacteria. Association of aerobic bacteria. Assume it as a aerobic bacteria. Because we read mitochondria help in the respiration in the aerobic conditions, and suppose this is your cyanobacteria. So after the association of the aerobic bacteria and cyanobacteria, what we got? We got the organelles. We got the organelles, that is the chloroplast, and the main, that is the mitochondria. mitochondria. And this is your eukaryotic ancestral eukaryote. What is that? Ancestral eukaryote. Prokaryote cell came from the eukaryotic cell came from the prokaryote. So जो हमारे माइटोकॉन्ड्रिया है और क्लोरोप्लास्ट है वो भी किससे बने? बैक्टीरिया से बने. कैसे बने हमारे ये? जब एरोबिक बैक्टीरिया और साइनोबैक्टीरिया ने हमारे एंसेस्ट्रल प्रोकैरियोट के साथ क्या किया? एसोसिएशन किया, तब हमारे पास क्या बना? एंसेस्ट्रल यूकैरियोट दैट कंटेन्स द ऑर्गेनेल्स, माइटोकॉन्ड्रिया एंड क्लोरोप्लास्ट. नाउ द क्वेश्चन दैट वी वर डिस्कसिंग दिस वन कि कैसे जो है हमारा नॉन न्यूक्लियर डीएनए मींस माइटोकॉन्ड्रियल डीएनए इनहेरिट होता है फ्रॉम द पेरेंट टू द प्रोजेनी. सो इसको हम क्या कहेंगे? दिस इज जेनेटिक पोर्शन बट लेट्स डिस्कस इन द ब्रीफ. दैट इज द साइटोप्लाज्मिक इनहेरिटेंस. व्हाट इज दैट? साइटोप्लाज्मिक inheritance or the maternal inheritance. First thing we need to remember that the copy number, there is a high copy number of double stranded circular DNA. कि जो माइटोकॉन्ड्रिया में सर्कुलर डबल स्ट्रैंडेड डीएनए होगा उसकी कॉपी नंबर क्या होगा? हाई होगा. फर्स्ट थिंग. एंड जब भी रैंडम सेग्रीगेशन, सेकेंड इज द सेग्रीगेशन. नाउ व्हाट इज द मीनिंग ऑफ द रैंडम सेग्रीगेशन? फॉर एग्जांपल इफ सेल वांट्स टू डिवाइड, okay, cell wants to divide, so there will be a lot of mitochondria in a cell and only one nucleus will be present. So जब भी सेल डिवाइड करेगा तो फॉर एग्जाम्पल 3 एक साइड चले गए, 3 एक साइड चले गए, बट न्यूक्लियस में क्या होता है कि प्रॉपर दैट इज दी कि हाफ ऑफ डिवीजन होगा उसमें. बट माइटोकॉन्ड्रिया में क्या हो रहा है? रैंडम सेग्रीगेशन हो रहा है, रैंडम डिस्ट्रीब्यूशन. So random distribution in the daughter cells during the mitosis and the meiosis, as well as the during fertilization. The main concept is here during fertilization. For example, we have this cell that we call it as the mature ovum. What is the number? N is the number. And this is the sperm N. Now mature ovum will have the mitochondria as well as mitochondria. Now you will say, ma'am, in sperm how it will move? It also requires the mitochondria. Yes, but when they will fuse during the fertilization, when they will fuse, so only the sperm will inherit its nuclear DNA. It will inherit only the nuclear DNA, not the cytoplasm. It will form the zygote, that is the diploid, and the zygote only contains the maternal mitochondria. If progeny faces or there is a problem in the genes that is related to the mitochondria, so we will only diagnose only the maternal side. हम पीछे जो मदर है और दादी नानी सब है उनके हम जीन्स को क्या करेंगे? एनालाइज करेंगे. And this mitochondrial inheritance is the single parent inheritance है. कैसे है? Because जो न्यूक्लियर जीन होंगे, न्यूक्लियर जीन्स that will comes from the both mother and father. But जो mitochondrial genes होंगे that only comes on from the mother, not from the father. So this is very important topic as well as in the graduation and post graduation. So this is all about the mitochondria. Now let's understand about the our second organelle that we have, chloroplast. Now chloroplast is present, it is a organelle. So जब हमारे पास organelle होगी that is the plastid. जो plastid का type है that is the different types होंगे, chloroplast, chromoplast, as the जो third वाली हमारे पास that is the leucoplast. Now this chloroplast is only present in the plant cell as well as the euglena. Present in plant cell as well as the euglena. Clear? As well as these plastids are easily observable under microscope. आप इसको easily microscope के अंदर easily देख सकते हो. Now these plastids are of different types. Now how the plants perform photosynthesis. So and at which light? So when we talk about कि हमारे पास क्या है, प्लांट जो है हर लाइट में फोटोसिंथेसिस नहीं करते हैं. हमारे पास क्या है, एक विजिबल लाइट है, विजिबल लाइट जिसके अंदर ही प्लांट क्या करेंगे, फोटोसिंथेसिस करेंगे. एंड दैट रेंजेस फ्रॉम दी 400 नैनोमीटर टू 700 नैनोमीटर. Yes, so one is the leucoplast, one is the chloroplast, one is the chromoplast. So leucoplast gives no color. What is that? It gives no color. It gives no pigment. Chloroplast gives green color. Chromoplast other than green color, orange red. Orange red means you can say other than green. Yes. Now the leucoplast involved in the storage. और जो chloroplast that helps in the photosynthesis. Now let's discuss about the chloroplast. हम discuss करेंगे chloroplast, why it is a autonomous organelle और क्या इसमें और special है. So chloroplast is a double membranous organelle. क्या है ये? Double membranous organelle है. Outer membrane and inner membrane. And this is the outer membrane. This one is your inner membrane. This is the intermembranous space. Now inside the matrix, what is present? I draw like this. are the stacks of what? Thylakoid. These are the thylakoid. And multiple thylakoid stacks on one another, we will call it as the granum. And multiple of granum, that is we call it as the lot of granum is present, plural grana. Yes. And this is the stalk like structure, that is the stroma. Stroma lemon. Ribosomes are present. And what kind of ribosomes? 70S ribosomes are present. And we already know plants store food in the form of the starch. So obviously starch will be also present. Starch granules. Now what is the significance of the thylakoid and stroma lamellae? So thylakoid is responsible for the light reaction, whereas this cytoplasm we call it as the stroma. It is responsible for the dark reaction. Now what is light reaction and dark reaction? Let's understand a little bit difference between. One, we have the light reaction. One, we have the dark reaction. So we discussed there is a thylakoid. So there is a membrane of the thylakoid. In the membrane of the thylakoid, what is present? This pigment is present, chloroplast is present. In this, chloroplast is present. They are arranged parallel to the membrane of the thylakoid. So these pigments, what kind of the pigments? Chlorophyll A, chlorophyll B, carotenes. That is the catenoids and xanthophylls. These are the accessory pigments. Chlorophyll A is the universal pigment, main pigment, universal pigment. Means these other pigment will absorb the sufficient amount of light and handovers to the chlorophyll A. Now chlorophyll A, what will happen? Electrons will excited during the absorption of the quanta, that is the sunlight, and they goes to higher electron state. So in this, what happens? The light reaction occurs in the thylakoid membrane, and it is dependent on the light. Now during light reaction, what will happen? There is a formation of the ATP plus NADPH. Now ATP and NADPH formed during the photosystem 1 and photosystem 2, means cyclic and non-cyclic photophosphorylation. Cyclic and non-cyclic photophosphorylation. Now what is the meaning of photophosphorylation? It is the formation of the ATP in the presence of the sunlight. So we have observed this structure here. So here non-oppressed part means non-sandwiched part. Here photosystem 1 will run, and here this is the oppressed part, means sandwiched part, photosystem 2 will be present, and this is non-oppressed part, photosystem 1 is present. And along with the photosystem 2, here what else things are present here? It is the NADPH reductase enzyme is also present. Okay. So due after the continuation of the photosystem, जब हमारा photosystem 1 और photosystem 2 चलेगा, उसके बाद हमारे पास क्या बनेगी, ATP और NADPH क्या बनेगा, जनरेट होगा. और डार्क रिएक्शन में क्या होगा, that occurs in the stroma, जो कहाँ पे चलेगा, in the stroma. Why? Because all the enzymes that is required for the dark reaction are present in the stroma में. Occurs in stroma. And what will happen? There is a reduction of the CO2. यहाँ से क्या होगा? Reduction of the CO2 होगा. And it will form the carbohydrate. क्या बनाएंगे हम? Carbohydrate that is in the form of glucose. And we will utilize the ATP plus NADPH for the generation of the carbohydrate that is the glucose. Now we have chlorophyll A and chlorophyll B pigment that is present. Chlorophyll A, chlorophyll B pigments are present. So there is only the difference between the chemical reaction that they have, and in these they have magnesium that is the center reaction, and 4N is present and bounded with the ring. So we will discuss this structure in the next class. I hope you like this session. Thank you.